Okay, so part 2 of week 5 of the SARS-CoV-2 lecture series is about vaccines, and we're going to look at the different vaccine types and ask the question, will they work? And if so, how do they work? Okay, so I said here, we don't know, because we only have relatively short-term data. We know that in the short period of time of a year or so, these vaccines work pretty well against severe disease and death, but they are not as good as we would want them to be at protecting individuals from just receiving an infectious, symptomatic infection. We know that in the short term, IgG levels, which are antibody, antibodies that you find within plasma and tissues, are maintained for 6 months at pretty high levels, and IgM, which is a different type of antibody, is maintained for longer. And we know that these antibodies can neutralize the virus in the test tube, which is one of the tests of how good the vaccines are. We take the patient blood and we test to see if those antibodies in blood can neutralize the virus and prevent it from being infectious. We also know that many patients have been infected twice within a period of 6 months from genetically different viruses. So some people were infected with, let's say, the alpha variant, the end of last year, and then we're reinfected with the Delta variant this year. So although we have seen some reinfections, we haven't seen anything like the number of reinfections that you would expect to see. So we know that short-term immunity exists. We know that that immunity can last for at least a year. We also know that it might wane after 6 months, so you might go from being 95% protected to maybe 93% protected. Now that sounds like a very small drop, but that is actually quite a substantial drop in immunity. So short-term immunity is very, very good. Longer term, we may get loss of immunity. And to understand why loss of immunity is likely to happen, all we have to do is look at other coronaviruses that cause common cold. And there were 3 main ones in circulation here. And what we see is the frequency of those over time. And what you can see is they tend to occur commonly maybe every other year or every 3 years. So that would suggest that there is protective herd immunity in the population, but then after a couple of years, after a big outbreak of it, in the first year, the majority of the population are immune to it, and then in the second or third year, herd immunity has decreased, and therefore the virus can then have another outbreak, and we see a spike of activity. So you can see a massive outbreak of this virus in 2010, and then levels were very, very low in 2011, 2012, and then it started to rise again in 2013. And then we have a few quiet years before it comes back again. So that's a classic situation where immunity is waning after a couple of years, and then it will come back. So if we look at the different vaccine types, and we're going to look at how they work and look at some of the data. And we've got various different approaches, some of which are completely new thanks to this pandemic. So we can have what we call live attenuated virus expressing the SARS-CoV-2 proteins in a non-pathogenic virus. This is what we get in AstraZeneca and Johnson & Johnson. So this is an unrelated virus that doesn't cause disease in humans, but you make that virus express the SARS-CoV-2 spike protein, and then those cells infect us, those viruses infect us, and then the infected cells produce the spike protein, and that's what we get our immunity against. I'll show you some of that later. We can have killed viral vaccines where a pure virus preparation is concentrated and then basically killed with an organic solvent so that they are no longer infectious. So you're injecting dead virus particles. There's viral protein nanoparticles, Novovax, which at the time of writing still not formally approved, but it works really well. And then we've got the viral RNA vaccines by Pfizer and Mrna. So firstly, we've got the Adenovirus, this is the Oxford AstraZeneca and Johnson & Johnson vaccines, and these use an adenovirus. So adenovirus is just one of the many viruses out there, and this particular virus can cause, or is related to viruses that can cause common cold, and different adenovirus strains are used in lots of different vaccines. So they're very well characterized. They've been used for loads and loads of vaccines, including flu vaccines. And in all of these vaccine types, we are just expressing the viral proteins from the pathogen on this very innocuous adenovirus, injecting that into patients. And this adenovirus has been made to be replication incompetent. So it can get into cells and express proteins, but what it can't do is then replicate within those cells to infect more cells and cause some sort of disease. So this adenovirus is expressing SARS-CoV-2 spike protein, and therefore every cell that this virus gets into, this adenovirus, it will express SARS-CoV-2 on the infected cell surface, and then you get immunity to that. And if you click on this link on the PowerPoint version of the slides, you'll see a video on the technology, and here you'll see the first study of this particular vaccine being put into primates, non-human primates. So you'll see just 5 months after the pandemic really got going, we've got this vaccine that was put into non-human primates, and showing that it will protect against SARS-CoV-2 in rhesus macaques. And this is just a bit of the abstract explaining the key things of the paper. And this is some of the first data that came out. This was published in December, so 5 months after the macaque study. We've got the finalised AstraZeneca vaccine going into humans. And this is how we express the data for a trial of this sort, where we have in orangey red, we've got the patients who received the vaccine, and in blue we have patients who have received a control vaccine for something completely unrelated. So they received a vaccine, just not against SARS-CoV-2. It was a meningococcal vaccine, completely unrelated. So in a way, this controls for some of the mild side effects that you might receive, you might expect to get when you have a vaccination, of flu-like symptoms and so on. So these in blue were not just receiving a placebo, a proper placebo like some saline injection, they're receiving a different vaccine. And what you can see is this is cumulative proportion of patients coming up with some sort of disease, reporting symptomatic COVID-19 disease, and you can see that the frequency in the vaccinated group is much lower than in the control vaccinated group. Now the legend's a little bit confusing here, because there were some patients who have a standard dose and some patients had a lower dose, and they combined those together. There was a bit of an error in the clinical trial. Some patients had the wrong dose, but they combined all the patients together. And the data's also got an exclusion period, which is the period from actual vaccination through to this period of a couple of weeks, because we know it takes a couple of weeks for the vaccines to work. So during this exclusion period, you would expect the vaccinated and the unvaccinated to start to be catching SARS-CoV-2 at a similar rate, and then the line should start to separate. So we just exclude that period because that can't really be related to the vaccine. So what we found with sort of more longer term data is we've got greater than 94% efficacy on hospitalizations and death, and a lower protection against infection. And this is from this later paper here. So the initial data was not quite as good as we expected, but then the follow-up studies were really very impressive. So that was the AstraZeneca vaccine. Then there's also the inactivated virus, and lots of vaccines are prepared in this way. And all you do is you culture the virus in this case, African green monkey kidney cells, just the type of cell that would grow in the lab. You get a purified viral preparation and then inactivate it with beta propionate and do that twice to kill off all of the virus, hopefully. And then you mix it with aluminium salts to act as what we call an adjuvant, which enhances the immune response. And this is basically the entire method of how to make a vaccine, which is the Sinovac, or the Chinese vaccine. So that's exactly how they made it. So any good scientist will be able to follow that exact method and make their own vaccine. It's really quite simple. And there's a link there to the article of the first trial using this data showing its efficacy. And this vaccine's had some really quite bad press in that in the clinical trials, this is a cumulative incidence. A lot of this was performed in Chile, South America. The unvaccinated group showed this particular rate of increase over the study period. Those that had vaccinated with 2 doses showed a much lower rate, but those vaccinated with only a single dose actually showed a higher rate of SARS-CoV-2 infection. And we don't know whether this is a behavioural effect where after a single dose, if the patients are not well educated, you're injected with the vaccine and then you immediately go out and think that you can ignore all social distancing norms and forget the fact that it's going to take a few weeks for the vaccine to have its benefit effects, but this effect seems to last long term. So a single dose, it's really quite inexplicable as to why this happened. And the final data that came out in terms of vaccine effectiveness against symptomatic disease was really not very good, but the fully immunised was pretty impressive. 86% protection against death after a double vaccination, 90% protection against intensive care admission, and 87% protection against hospitalisation. So a double vaccinated regime works really pretty well, as well as most vaccines do. It's just inexplicable as to why the partly vaccinated seem to do worse. However, there is an issue here as this in Chile. Chile's got one of the highest rates of SARS-CoV-2 in the world, the highest death rate in the world, and whether there is some effects going on there where part of the trial was done in an area with low SARS-CoV-2 rates, which then really took off mid-trial, which might have affected the data. The other possibility is whether people react differently if they've recently been infected with SARS-CoV-2. So maybe it's because SARS-CoV-2 was at such a ridiculously high rate in the country while this trial was going on. That is possible, because I mean, Chile is one of those countries where up to 0.3% of the population has died from this infection. That's 0.3% of the entire population. About 1 in 300 people have died of this. So there may be something there that is messing up the trials. Okay, third viral type, vaccine type is the Novovax spike protein nanoparticles. As far as I know, this has still not been formally approved in the UK, but the trials of it were very impressive, giving really good protection. And this is a completely novel way of delivering vaccines. Okay, so I'm not going to play the video here, but you can go to the PowerPoint version of the slides, click on here, and you will get this short video explaining how these SARS-CoV-2 spike protein nanoparticles are made. But to cut a long story short, we engineer the spike protein to be expressed as a little cluster of proteins, and then we purify those and inject them. And these clusters of spike protein are about the same size as the virus and elicit a very similar immune response to an intact virus. So watch that video and you will see how those vaccines are made. Suffice to say, the data is very, very good. So this is a placebo. So this is someone injected with something like saline, such as sodium chloride solution, versus those infected with the Novovax, and the cumulative incidence. And you can see that those two lines separate very quickly after the 7 days after the second dose was given, which is the exclusion period of time. So you can see that the cumulative incidence is about 0.2% versus up here approaching 2%. So we're looking at about 90% efficacy. and that's against both hospitalization and death. So finally, we're onto the mRNA vaccines, and the main ones are the Moderna Therapeutics and the Pfizer vaccines. So these vaccines have been quite controversial because they are new, completely new ways of delivering vaccines that have not been done routinely previously. Now, the technology has been applied for other vaccination situations, particularly trying to create cancer vaccines, trying to initiate patients who've got cancer to have an immune reaction against their own cancers. And that's where the Pfizer-BioNTech vaccine technology had been previously trialled. So this idea, we've been putting these mRNA vaccines into patients for years, just not in any great numbers. So the way this works, you take the viral messenger RNA that encodes the spike protein and encase that in a lipid coat, and that lipid coat allows the mRNA to be taken up by cells. We then inject that into the patient, so wherever those lipid droplets that are protecting that mRNA go, they'll be taken up locally by muscle cells, and those muscle cells will translate the mRNA to produce spike protein, which then gets expressed on the surface of those muscle cells. The immune cells come along and recognize that the spike protein is present on the surface of the cells and recognize that as foreign. Now, sometimes that can be intact spike protein, sometimes it can be presented. The protein can be chopped up by the cell and presented as peptide fragments. Either way, the cell is expressing something foreign that the immune system has never seen before. So the immune system comes along, recognizes those infected cells, destroys those infected cells, but recognizes the spike protein, and then activates the acquired immune system to encourage B cells to make antibodies that recognize SARS-CoV-2 spike protein, and T cells that will also seek out and destroy infected cells. So there's a really nice article here explaining how this technology works. And the data is pretty spectacular. Again, this is cumulative incidence of placebo versus the Pfizer-BioNTech vaccine. And you can see in days after dose 1, this is just after one dose, you can see that after about 10 days, these two lines diverge. We continue to get patients getting infected in the placebo arm, whereas these patients are pretty much protected, and there are very, very few severe cases. Most of the cases in the vaccine arm are much milder cases than in the placebo arm. So we've got long-term data showing sustained reductions in hospitalizations and deaths, and we've got very similar data for the Moderna vaccine, which works in a very similar way. Now, this is fairly old data now, but showing just how effective the vaccines are. This is looking at data from April to August, April the 1st to August the 18th of 2021. And what we can see is that in countries where we've got vaccination, we've still got really quite substantial high case rates, but what we're not getting is the corresponding death. Whereas in countries where vaccination is at a very low level, where we get a spike in cases, we get a spike in deaths. So the vaccine seems to have helped uncouple cases leading to hospitalizations and deaths. Now, there are some co-founders here that makes this data probably look better than it really is. If we think back just to the UK, we had a massive spike in the Euros when the Euro football matches were on, and a lot of people who got that infection were really quite young then. So this data is biased towards younger people catching the infection. But even when we look at the older people who subsequently catch the infection, we have got quite really very substantial protection from death, and we can be pretty sure that this would have been, down here, this red deaths part of the graph. And here's some nice data showing various different situations from September 2021, looking at COVID-19 cases presenting in emergency care, looking at the vaccinated with at least 2 doses versus the unvaccinated, and this axis here is rates per 100,000 people. So we know that if you are vaccinated, this is the degree of protection that you are receiving. So you'll see some statistics at the moment kicking around that will say that there are the vast majority of people who died due to COVID-19 in the last year have been double vaccinated. Well, that is true. That's because the vast majority of the population has been vaccinated. When you correct for case rates per 100,000 in each group, you'll see the degree of protection that you are getting. So this is presenting to emergency care within 28 days of a specimen vaccinated versus unvaccinated. We've got death within 28 days of a positive test, vaccinated versus unvaccinated, and also death within 60 days of a positive test, vaccinated, unvaccinated. And yes, this bar is this is bigger than we wanted it to be, but it is still a really substantial decrease in death for that older age group. And here is probably the most controversial graph that is doing the rounds at the moment, particularly forwarded on by anti-vax groups. And this is a graph which is using government data. It's not the most reliable government data, but it is government data, and it actually shows the infection rate as opposed to whether you're vaccinated versus unvaccinated. And it actually gives the impression that those vaccinated twice might have a higher rate of SARS-CoV-2 infection than those who are unvaccinated. Now, the reason why this is so controversial is this data has been put together by the Office of National Statistics and and the government, so it's coming from the same sort of place as here. So it's government data, not Office of National Statistics, government data, and it is looking at vaccine versus unvaccinated, looking at rates. So it looks like it should all be controlled for. The problem with this data is we don't really know how many people there are in the country, and we don't really know what proportion have really been vaccinated. And if we're up at the point where 90% of the population are vaccinated in a particular age group, and the error of how many people we have in the country in that particular age group has got an error of a few percent, then you can get quirks in the data that suggest that there are those who have been vaccinated are doing worse than those who are unvaccinated. So it's a problem with statistics, and although it is clear that the vaccines are not protecting against infection anything like as well as we thought it was, the general consensus is that this graph is a statistical issue, and if you use a different estimate of the current population, these bars are reversed. But it's a very controversial area. I don't know which is correct, but well, I suspect that this is the least reliable of the data sets. But certainly the vaccines are not anywhere near 100% effective at preventing infections. Now, whether those people who are vaccinated are getting infections but are not as effective, there is strong evidence to suggest that that is true. So we know if you're being vaccinated, you're less likely to die, less likely to be hospitalised. We think you're less likely to be infectious, but it doesn't exclude you from being infectious. And we think that if you are infectious, you become non-infectious much quicker than if you are unvaccinated. So when you see headline data of saying that vaccinated and unvaccinated have a similar rate of positivity, then there is some critical analysis of the data that requires doing. And that's as much as I will say. Okay, that's the end of this session on vaccines and their effectiveness or not.